Okay, in this video, I'm going to draw for you the life cycle of a typical member of the Basidiomycota, and these are your club fungi. They include most of the things that we think of as mushrooms, and a lot of other fungi as well, being one of our two largest phyla. And the thing that characterizes a member of this phylum is a structure with 4 spores on it called a basidium. This is a basidium. It looks kind of like a club, and the basidium produces these things, and these things are basidiospores. And the fact that there are 4 of them gives you a clue as to how they are produced. But let's take one of these basidiospores and germinate it to form a hypha. Now, basidiomycetes have septate hyphae, which means that there are cross walls between each nucleus. So the nuclei are separated by a cross wall. So in this case, there's only one nucleus in each chamber, or each cell. So you can say this particular hypha here is monokaryotic. One nucleus. So what often happens in the basidiomycetes is that 2 monokarya may fuse together to form, can you guess before I draw it? What we call a dikaryon. So they have 2 nuclei per cell. And these dikaryotic hyphae can persist for a very long time. Very long time. So what's happening here in this junction is plasmogamy, or fusion of the plasma membranes of 2 cells. And this dikaryotic hypha, we describe it not as being truly haploid or diploid. We refer to it as being N plus N, is how we indicate that it's dikaryotic. So both of these nuclei are acting independently, transcribing and translating proteins. They may be genetically distinct from each other. That's the typical case. Maybe they're genetically identical, is less often the case. But these dikaryons can exist for a very long time before they do anything else. So what's the next thing they're going to do eventually is they may form into a large mycelium, and they may produce something that looks kind of like this. And this thing here, you might recognize it as a mushroom, but the technical name for it is a basidiocarp. And a basidiocarp has a cap called a pileus, and it's got these gills that are called lamellae. And the stalk of a mushroom, or what you might have thought of as the stem, is technically a stipe. S-T-I-P-E. And many mushrooms, but not all of them, have a ring-like structure called an annulus. And that's your typical mushroom. So the basidiocarp, or mushroom, has on these lamellae if we were to cut a lamella, so this is like a cross section of a lamella, and we looked under the microscope, we'd see it has lots and lots of little basidia on it. This is where the basidia are produced. And these things are teeny tiny, microscopically small. But if we were to zoom in on that, before we have a full basidium, we'd see that we've got a dikaryotic probasidium, and what happens next is those two nuclei fuse, so karyogamy is going to happen. It's going to give us a zygote, and because these are fungi that undergo zygotic meiosis, the next thing that's going to happen is meiosis. Which gives us 4 cells. Those cells migrate to the outside of the basidium, and that's how we get our basidiospores. That's why there are 4 of them, because they are the products of meiosis. So that is your generalized life cycle of a Basidiomycete mushroom in the Basidiomycota.